All right, good morning. So I'm going to go ahead and run through this meiosis presentation real quick, and then I'll stop recording. And then after that, we can talk about anything else. This won't take too long. All right, so last week I recorded a presentation on the cell cycle and mitosis. So if you haven't watched that, definitely recommend going back and watching that. It's pretty quick. So as kind of a quick recap, the cell cycle consists of interphase, which is G1S to G2. Basically, it's just where cells spend most of their life, and they start preparing for cell division, and the DNA replicates. Then they go through mitosis, which consists of prophase, metaphase, anaphase, telophase, which is just how the nucleus divides in half. So mitosis is just, it's cell division for most cells, for most body cells. It creates two new cells. They're genetically identical. They're for normal growth and repair. Okay, normally when you think of cell division, you kind of think of mitosis. Meiosis is slightly different in that it's cell division for sex cells, or what we call gametes. Gametes is just a fancy word for sex cell, like sperm and egg. Now, gametes are different from body cells or somatic cells in mitosis in that they're haploid. So think of haploid as half, because they have half the number of chromosomes as a typical body cell, which makes sense because, if we're talking about sperm and egg, you get half your genetic material from each of your parents. So they combine, that makes a body cell, that makes a full set of chromosomes, which is why half of your DNA is from each of your parents. All right, and they kind of along the same line. So you might have some questions that ask about the chromosome numbers. So for example, if you have, I don't know, I'm just making stuff up here. Well, here, we'll do a real one. So in humans, you have 46 chromosomes in every single one of your cells, every single one of your body cells. That means in gametes or sex cells, they're only going to have half of that. So they're going to have 23. So sex cells are going to have half or 23. Now, your exam, it could ask you about any other organism. It'll tell you a number. You just divide it in half to get the number of sex cells or chromosomes. Or you could be given the reverse. You could be given the sex cell, and then you just double the number of chromosomes to go to the body cell. So make sure you know how to do that. You're either doubling it or dividing in half. So again, back to meiosis. It's very similar to mitosis in that it's replicating cells, replicating the nucleus, except the major difference here, like I said, different type of cells. You end up with 4 cells instead of 2 new cells. One way I usually have students remember, so in meiosis, think of the E and S in meiosis as egg and sperm. So egg and sperm cells. So pretty much anything dealing with reproduction is going to be meiosis. Mitosis, everything else, normal growth, repair. All right, so all the steps of meiosis are fairly similar, except they occur twice. I'm only going to touch on the differences. I don't think you'll be asked about specific phases of meiosis because it's so similar to mitosis. All right, so this is the first change that you'll definitely be asked about on at least your end of course exam, which is really important. So crossing over occurs during, we call it prophase 1 because it goes through everything twice. So there's prophase 1, metaphase 1, anaphase 1, telophase 1, and then you go to prophase 2, metaphase 2, anaphase 2, telophase 2. Just kind of keep that in mind. So during prophase 1, this is crossing over is what causes the genetic diversity. This is what causes the cells to become genetically different, as opposed to mitosis, where they're just the same exact cells. If you get a cut or whatever on your skin, you need new cells. Of course, you want them to be the same. Whereas genetic variation is important for reproduction. It's one of the huge benefits of it. So yeah, so that process is called crossing over. It leads to the independent assortment of chromosomes. It's just a fancy way of mixing up or the DNA, causing genetic variation. All right, those are the steps. Again, I'm not going to go into those in too much detail. That's part 2. The only thing I want to point out, so step 1, you end up with 2 cells, and then those 2 cells are going to go through the process again. You end up with 4 cells. So, just make sure 4 cells after meiosis, only 2 cells after mitosis. All right, polar body. I don't know if this will be on your end of course exam, but it'll probably be on your exam in this class. So when egg cells are created, so egg cells are created during meiosis, you end up with 4 cells. However, only one of those cells is actually usable. So even though there's 4 cells made, only one cell is actually usable. So the others are what we call polar bodies. They don't really have much of a function. Whereas in sperm cells, there's 4 made, and that's that. They're all used. So it's more about in egg cells, it's more about quality rather than quantity. In sperm cells, it's about quantity rather than quality. All right. Okay, kind of same thing here, similar idea. So there's sexual reproduction, asexual reproduction. Make sure you know the difference, but it's pretty self-explanatory. So sexual reproduction, 2 parents gonna provide unique, genetically different DNA. And this process is a little bit slower, but that's the huge benefit. Genetic variation is huge. It allows organisms to adapt. Um, so that's really important. Asexual, okay, you only need one parent. It's the exact same DNA. It's the same thing as making a clone of itself. So this is a lot quicker, but generally there's no genetic variation unless there's something else that influences the DNA, like a mutation, but we'll get into that in another talk. All right, and that's it. I'm gonna go ahead and stop this recording.